All right, in this presentation, I'll be discussing the ability of naturally derived oils and disinfectants to inactivate SARS CoV-2. First is to establish a bit of an introduction and precedence. Essential oils are oils that are derived from plant material, and usually by steam distillation, and they can come from different parts of the plant, from the root to leaves or bark. And they contain several volatile molecules. A single oil can contain as many as 20 to 100 different compounds, but the most common are terpenes, specifically monoterpenes and sesquiterpenes. These are organic molecules that can be quite volatile. There is quite a bit of precedence for previous work in viruses being susceptible to essential oils in the literature. There have been several different tests using various essential oils and various viruses as well as bacteria. A few of the viruses that have been well documented in literature include human herpes virus, influenza virus, Coxsackie virus, and coronaviruses. There's a lot of research going on right now to determine the specific mechanisms that the essential oils and their molecules use to inactivate viruses. And specifically, they tend to mask binding domains, which prevents the virus from entering a cell. They can also degrade the capsid, which also can kind of distort those binding domains as well as destroy the viral particle. And also it can adjust the cellular and endosomal pH, which affects whether the virus is able to decapsulate or be released into the cell. And there's also sometimes changes in the targeting genome, as well as interfering with signaling pathways within the cell and within the virion. There is a fair amount of background research into essential oils being used against SARS CoV-2. As we all know, this pandemic has changed the world, and there's been quite a few researchers that have been trying to identify whether essential oils are viable disinfectants against SARS CoV-2. In 2022, there was a researcher that discovered that tea tree oil is effective against two surrogate viruses, which included a feline coronavirus and a human coronavirus. These were both not as pathogenic as SARS CoV-2, but they worked as a good model. And just recently, a research study was published which showed that thyme oil was effective against SARS CoV-2 in vapor phase. And then in 2021, a researcher identified that several oils, including lemon oil, geranium oil, citronelle oil, and others, have been studied for clinical applications against the viral spike protein in silico and in vitro. That wasn't used as disinfectant, but that was used as a clinical study in that case. And there are several others in addition to these, but each were performed with different essential oils and different study conditions. In our study, we are using 4 different disinfectants. 2 of them contain disinfectant 1 and disinfectant 2 contain essential oils as well as other more standard disinfectants, including denatured alcohol or alkyl polyglucoside, and other standard disinfectants. And then disinfectants 3 and 4 are simply a combination of essential oils. And disinfectant 3 is the base oil that is found in disinfectant 1 and disinfectant 2. But all of our oils were blends of several different oils, meaning they contained many different compounds. In our procedure, we performed a basic disc-based carrier assay, which is where you dry the virus on a glass carrier and then add disinfectant for a set amount of time. And then as soon as that time is up, add a neutralizer to stop that disinfectant to calculate exactly how many viral particles remained after that disinfection process. And then we diluted it and used it to infect cells. And after that, we would incubate it for a period of 3 days and perform a plaque assay to analyze our data. Post-experiment, we took care of a few other things. So we would perform a plaque assay, which is used to calculate the number of plaque-forming units, which is equivalent to the number of viral particles that were used per carrier or per disc that we used. And then we also replicated our results. So all of our trials, we resulted over several days with 3 replicates on each of those days. And after that, we performed a statistical analysis, calculated our percent and log reduction, as well as our standard error, our p-value, and our r-squared, so that we could analyze our results in our data. This is an example of a plaque assay. So you have a very thin layer of cells, and as a viral particle destroys a region of cells, it leaves a little hole, and we count those little holes after the 72 hours, and they equivalent to the number of viral particles that the layer of cells was inoculated with. All right, some of our results. So for disinfectant 1, we had, let me see if I can get this out of the way here. So disinfectant 1, we had, we saw a significant log reduction within about a minute. So we calculated using 3 time points. We used 15 seconds, 30 seconds, and 60 seconds. And in that time, we were able to generate a prediction, a curve to predict where it would reach a 4 log reduction or 99.99% kill, which is what is necessary for significant disinfectant products by the CDC. And for disinfectant 1, it's predicted that for 4 log reduction, it would be 117.95 seconds for 4 log reduction or total kill, approximately total kill. And for disinfectant 2, we were also able to generate a curve. We used 30 seconds, 2 minutes, and 3 minutes. And with both of these, we noticed that if we went any longer than that last time point, we would have significant variation, and it was very difficult to calculate our curve and evaluate our data. For disinfectant 2, our predicted time for 4 log reduction was 8.64 minutes to get a 99.99% kill. For disinfectants 3 and 4, we tried various time points, but at 15 minutes, it still had less than a 1 log reduction for both of these disinfectants. And any longer than that, we determined to be impractical for use as a disinfectant. So even if we left it for 30 minutes and we saw more of a reduction, it wouldn't be practical to have a surface wet with that disinfectant for 30 minutes in order to have disinfection properties. So for these ones, we performed our replicates at 15 minutes and found that they were fairly consistent to between 0.7 and 0.9 log reductions. Some of our conclusions. From this data, we can deduce and predict that disinfectant 1 could be produced and replicated to be an effective disinfectant against SARS CoV-2, as long as it has a contact time of 117.95 seconds. And disinfectant 2 could also be used as an effective disinfectant against SARS CoV-2, with contact time of 8.64 minutes. And the results for disinfectants 3 and 4 do not indicate that they are effective against SARS CoV-2 as tested. And we do not have enough information to know if viral reduction in disinfectants 1 and 2 is caused by the essential oils or other ingredients. Remember, disinfectants 1 and 2, they contained other ingredients as well as the essential oils. Disinfectants 3 and 4 were the only ones that contained pure essential oils. Some ideas for future steps. Well, what we can do is identify the reactive chemicals in the essential oils and compare their effects to that of the pure oil. We can also compare the effects of other active ingredients in disinfectants 1 and 2 to the effects of the oils. So compare their oils versus the alcohol or the polyglucoside that were in those other 2 disinfectants. And we can also investigate other essential oils and blends to identify other potential disinfectants. One of the issues with essential oils is that they are so complex. There are that many compounds within it. And research has also shown that at times the isolated compounds perform better or worse than the oils themselves. And that they can vary significantly between growing season, as well as between what part of the plant the oil is being extracted from. And so these are some areas of research that might be able to help us better identify whether essential oils could be a good candidate for disinfection, or whether the ones that we chose were not the correct ones, or whether essential oils have any chance at being effective against SARS CoV-2. And just a quick acknowledgement that this project received funding and products from Young Living Essential Oils. And the products we used were the Thieves Spray, the Thieves Household Cleaner, and Thieves Essential Oil Blend and Purification Essential Oil Blend, which correspond to our 4 disinfectants. Here are our references. And thank you for your time.